Hi, my name is Angel. Welcome to MCAT Biochemistry. Today we are going to learn about enzyme structure and functions. This is going to be based off AMC 5th edition, right? So this is provided by Angel Loves Human Medicine, a great YouTube channel. So please like and subscribe. Comment on this video and let me know what you think about it. All right, so what are enzymes? Enzymes are proteins that help speed up metabolism or the chemical reactions in our bodies. They build some substances and break others down. All living things have enzymes, right? So they're responsible for building some substance and also for breaking others down. Our bodies naturally produce enzymes, but enzymes are also manufactured in products and food. So our bodies naturally produce them, but at the same time, enzymes are also produced in products and food, and you can consume them. All right. So what do enzymes do? Enzymes aid in digestion. They break down fats, proteins, and carbs. Enzymes use these nutrients for growth and cell repair. It also helps with breathing, building muscles, nerve functions, ridding our bodies of toxins, right? So it also helps with detoxifying everything that we don't need and or they are harmful to our bodies. All right, so what is the classification of enzyme, right? The group of enzyme oxidoreductases, transfer of hydrogen or oxygen atoms or electrons from one substrate to another. That's what they do. What is the example? Dehydrogenases, oxidases, transferases. Now that is a group of enzymes, right? They're responsible for transferring of specific group of phosphate or methyl, etc., from one substrate to another. Biggest example is transaminase and kinases. Another group is hydrolysis of a substrate. Esterases, digestive enzyme. Isomerases, change of the molecular form of the substrate. Phosphohexoisomerase and fumarase. Group of enzymes. Lysis, non-hydrolytic removal of a group or addition of a group to a substrate. Decarboxylases and aldolases are lysis, right? So what they do is lyse. Whenever you hear the word lyse, it's usually cutting off something off, right? Isomerases you have to remember as a change of molecular form of substrate. You know, the molecular formula changes, right? Hydrolases are nothing but the hydrolyze of substrate. Transferases, the name speaks for itself, a transfer of a specific group, a phosphate or methyl, from one substrate to another. Oxidoreductases, what is oxidation? Remember that. Transfer of hydrogen, oxygen atoms, or electrons from one substrate to another. Do not mix that with transferases, right? Ligases, synthetases, joining of two molecules by the formation of new bonds, right? Ligases joining. Citric acid synthetase is the example. How do temperature and pH affect enzymes? Enzymes need the right conditions to work, right? If conditions aren't right, enzymes can change shape. Then they no longer fit with substrate, so they don't work correctly. Each enzyme has an ideal temp and pH, right? So if it doesn't fit with the substrate, it cannot work, right? So shape defines its function. It's the same for enzyme. Enzyme has to have an ideal temperature and pH to function well. Now, enzymes are sensitive to acidity and alkalinity. They don't work properly if an environment is too acidic or basic. For example, an enzyme in the stomach called pepsin breaks down proteins. If your stomach doesn't have enough acid, pepsin cannot function optimally. That's right. We all need pepsin. Temperature, and that's where Pepsi comes from. It comes from pepsin, right? The famous drink. Um, Temperature enzymes work best when your body temp is normal, about 98.6 or 37 degrees Celsius. As temperature increases, enzyme reaction increase, but if the temperature gets too high, the enzyme stops working. That's why a higher fever can disrupt bodily function, right? It can cause serious issues that includes paralysis, that includes seizures. Most of the times when fever kind of increases, it causes some serious hysteria as well. Right, so anything that increases, when temperature increases, it has an impact on your pH, and a little bit of, change in pH can have some serious clinical issues in human body. So it's very important that you make sure that you maintain your pH and your temperature, right? That's why we hear a lot many bad news about folks dying due to hot weather or cold weather. Too much cold can disrupt body functions as well. Functions of enzymes in catalyzing biological reactions. Enzymes catalyze chemical reactions by lowering activation energy barriers and converting substrate molecules to products, right? They catalyze chemical reaction by lowering activation energy required to activate a particular chemical reaction, converting substrate molecules to products. A plus B equals to C plus D, right? So it helps lower the activation energy. Enzymes bind with chemical reactants called substrates, right? So chemical reactants are substrate. A plus B equals to C plus D. So A plus B is a chemical reactant, which we are going to call substrate going forward. A specific chemical substrate matches a site like a jigsaw puzzle, right? You've solved jigsaw puzzles when we were kids. I've done it and I loved it, right? So chemical substrate matches the sites as a perfect puzzle, and it sets up and makes the enzyme specific to its substrate. When an enzyme binds to its substrate, it forms an enzyme substrate complex. Very important. You'll hear me saying, excuse me, enzyme ES complex. That is enzyme substrate complex. After enzyme binds with the substrate, it is ES. This complex lowers activation energy of the reaction, promotes its rapid progression by providing certain ions or chemical groups that form covalent bonds with molecules as a necessary step of the reaction process. So the ES complex lowers the activation energy of the reaction, promotes its rapid progression. It increases the speed by providing certain ions or chemical groups that form covalent bonds, right, with molecules as a necessary step of the reaction process. So what it's doing is it's helping bind covalent bonds, only to make sure that the the reaction is speed up. So it's helping with progression. Promotes enzyme chemical reactions by bringing substrates together in optimal orientation, lining up the atoms and bonds of other molecule, which contours the substrate molecules and facilitates bond breaking, right? So it brings in the substrates together in an optimal orientation, lining up the atoms and bonds of the other molecule, which contours, it twists the substrate molecule and facilitate bond breaking. So it can help you break the bond, it just goes click off, right? That's what. The active side of an enzyme also creates an ideal environment, right? So you create a space, you have an ideal environment, slightly acidic or nonpolar environment for the reaction to occur. All right, so it's slightly acidic or nonpolar for the reaction to occur. The enzyme will always return to its original state at the completion of the reaction. So it comes back, it knows where it was, and it comes back. One of the important properties of enzyme is that they remain ultimately unchanged by the reactions that catalyze. That is a very high yield statement for MCAT, So enzyme is that it helps as a catalyst, but then it comes back to its normal location, right? So one of the important properties is that it remains ultimately unchanged. Nothing happens to the enzyme by the reaction they catalyze. All right, so there is no difference in the enzyme. They may have lowered the activation energy of the chemical reaction, but nothing happened to the enzyme. So they're foolproof. After an enzyme is done catalyzing a reaction, it releases its products, substrate. So after it's done, it releases them. Reduction of activation energy. Activation energy is the minimum amount of energy that must be available to reactants for a chemical reaction to occur. The minimum amount of energy that must be available to reactants for a chemical reaction to occur is called as activation energy. The activation energy EA of a reaction is measured in kilojoules per mole or kilocalories per mole. Activation energy can be thought of as a magnitude of potential barrier, sometimes called the energy barrier, separating minima of potential energy surface pertaining to the initial and the final thermodynamic state. So what was the initial state and what was the thermodynamic final state? So where was the activation energy reached and where did it subside? So that's what it decides, okay? So we use the Arrhenius equation, to calculate activation energy. And remember me when I say this is a very high yield topic, enzyme inhibition, competition, activation energy, they're all high yield topics. So you have no other option but to learn this. So you will have to learn it. A is the pre-exponential factor for the reaction. R is the universal gas constant. T is the absolute temperature, usually in kelvins. K is reaction rate of coefficient. Uh, Even without knowing A, EA can be evaluated from the variation reaction rate coefficient as a function of temp within the validity of Arrhenius equation. So there are some rules to it, but even if you don't know A or EA, you know A is the pre-exponential factor for the reaction. EA being the activation energy, you can certainly find out by the variation rate of coefficient. How variable was the coefficient rate? So you'll find out what the real Arrhenius equation is all about. How do enzymes speed up biochemical reactions so dramatically? So what is it about enzyme that helps us, right? Sometimes we wonder, why are you even helping us? Duh. Okay, so like all catalysts, enzymes work by lowering the activation energy of chemical reaction. Activation energy is the energy needed to start a chemical reaction, right? The biochemical reaction shown in the figure requires about 3 times as much of activation energy without the enzyme as it does with the enzyme. All right, so you know, so with the enzyme, like I said, it lowers activation energy. Without the enzyme, it will be like 3 times more activation energy. So enzymes helps you, right? All right, so the reaction represented in this graph is a combustion reaction involving the reactants glucose, C6H12O6, and oxygen O2. The products of the reaction are carbon dioxide and water. Energy is also released during the reaction. The enzyme speeds up the reaction by lowering the activation energy needed for the reaction to start. Compare the activation energy with and without the enzyme, right? So this is without the enzyme, this is with the enzyme. So without enzyme, you see how much of an activation energy was needed. This is with the enzyme, it has lowered the activation energy. The reactants are glucose plus oxygen, right? Overall energy during reaction product CO2 plus H2O. So this is the overall energy, but here as you can see, the activation energy is energy to activate a particular chemical reaction, right, is reduced. Okay, so you can compare and see the difference. Enzymes generally lower the activation energy, reducing the energy needed for reactants to come together and react, right? So these two are substrates, these are active site enzyme, ES complex, the product, right? So there's a new product that was created, and this is the enzyme, right? Enzyme brings reactions together, so they don't have to expend energy moving about until they collide at random. Enzymes bind both reactive molecules called the substrates tightly and specifically at a site on the enzyme's molecule called the active site. By binding reactants at the active site, enzymes also position reactants correctly, so they do not have to overcome intermolecular forces that would otherwise push them apart. So it's not only helping, helping lower the activation energy, but you also have to see they're helping them to stay at one place. So there is no steric pull, between two molecules. This allows molecules to interact with less energy. So the molecules interact with each other, but there is not a lot of energy that's been expended. All right, enzymes may also allow reactions to occur by different pathways that have lower activation energy. Active site is specific for the reactants of the biochemical reaction the enzymes catalyze, like puzzle pieces fitting together. Active sites can only bind certain substrates, so it's very specific. It's not for every molecule, right? So like puzzle pieces fitting to that together, active site can only bind certain substrates. Very picky, it's very specific, it's not all. So let us discuss about the substrates and enzyme specificity. Enzymes bind with chemical reactants called substrates, right? There may be one or more substrates for each type of enzyme, depending on the chemical reaction. In some reactions, a single reactant substrate is broken down into multiple products. In others, two substrates may come together to create one large molecule. Two reactants might also enter a reaction, both become modified, and then leave the reaction as two products, right? So two reactants might also enter a reaction, both become modified, and then leave the reaction as two products, right? So in some reactions, however, a single reactant substrate is broken down into multiple products, right? So one can be changed into two, and two can be changed into two products, right? So that is possibility. Enzymes active site binds to the substrate. Since enzymes are proteins, the site is composed of a unique combination of amino acid residues, side chain or R groups. Each amino acid residues can be large or small, weakly acidic or basic, hydrophilic or hydrophobic, and positively charged, negatively charged, or neutral. The position, sequences, structures, and properties of these residues create a very specific chemical environment within the active site. A specific chemical substrate matches this site like a jigsaw puzzle piece and makes the enzyme specific to its substrates, right? So it's not for everything, it's usually very specific to a particular substrate. Active sites and environmental conditions. Environmental conditions can affect an enzyme's active site and therefore the rate at which a chemical reaction can proceed. Increase the environmental temperature generally increase the reaction rates because the molecules are moving more quickly and are more likely to contact with each other. However, increasing or decreasing the temperature outside of an optimal range can affect chemical bonds with the enzyme and also change its shapes. So, increasing or decreasing temperature, hot or cold, can also impact the chemical bonds within the enzyme. If the enzyme changes shape, the active site may no longer bind to the appropriate substrate and the rate of reaction will decrease. It may not fit in very well. Dramatic changes to the temp and pH will eventually cause enzymes to denature. So, like we talked about the thermodynamic stage in the initial of final, the temperature and pH, which eventually will denature the enzyme if they're not managed. And if there is a drastic change to it, if it gets really hot, then it will denature the enzyme. Induced fit and enzyme function asserted that enzyme and substrate fit together perfectly in one instantaneous step. As the enzyme and substrate come together, their interaction causes a mild shift in the enzyme structure that confirms an ideal binding arrangement between enzyme and its substrate. The dynamic binding maximizes the enzyme's ability to catalyze its reaction, right? So what is the awesome thing about induced fit and enzyme function? The dynamic binding maximizes the enzyme's activity. So the function improvises because of the dynamic binding and ability to catalyze its reaction. Asserted that the enzyme and substrate fit together perfectly in one and instantaneous step, right? So it's like a click flooring. It clicks on its own, right? Perfectly. Induced fit model. According to Induced fit model, both enzyme and substrate undergo dynamic conformational changes upon binding. Enzyme contours the substrate, it twists into its transition state, thereby increasing the rate of the reaction. So this is the substrate. This is the active site. This particular site is an active site. Substrate entering the active site. It's entering active site of the enzyme. Now they form an enzyme substrate complex, the ES complex. Enzyme changes the shape slightly as substrate binds. So it's changing it slightly. As you can see, it was a little narrow and now it got a little broad, right? So enzyme product complex. So this is the product. This is the complex that they build. This is the product. It's perfect, right? It's fitted very well. The shape is very good. And now the products are ready to be released. Products leaving active site of enzyme is released. So induced fit model is more like a mold for clay. You know, when you're kids, you must be playing with clay, where you put it in a round or square, and it takes a perfect shape. When it's ready, we take it off, and we build whatever we are trying to build. I used to build teddy bears, but I'm just saying, right? So these are the products leaving. The enzyme's active site binds to the substrate, so remember that. So active site binds to the substrate, increasing the temp, generally increasing the rate of a reaction, but dramatic changes in temp and pH can denature an enzyme, thereby abolishing its active action as a catalyst. The induced fit model states a substrate binds to an active site and both change shape slightly, creating an ideal fit for catalysis. When an enzyme binds its substrate, it forms an enzyme substrate complex. Enzymes promote chemical reactions by bringing substrates together in an optimal orientation, thus creating an ideal chemical environment for the reaction to occur. So enzyme and idealization kind of goes, hand in hand. So wherever you have enzymes, you'll see the environment is a little more idealistic than without the enzyme. And this is exactly why it's three times more, it requires three times more activation energy without enzyme. The enzyme will always return to its original state at the completion of the reaction. So after the products are done, enzyme will go back to its normal state. It does not change, it doesn't get utilized, it goes back to normal, right? Active site model. What is active site model, right? We know about active sites. Describe the part of an enzyme to which the substrate binds and where a reaction is catalyzed. The enzyme's active site binds to the substrate. So this active site, the enzyme's active site binds to the substrate. Since enzymes are proteins, the site is composed of a unique combination of amino acid residues, side chain or R groups, right? So this is substrate, this active site, enzyme, ES complex is created. So as you can see, there is no two, you don't see two reactants coming together. It's only one particular substrate we are talking about. Part of an enzyme to which substrates bind where reaction is catalyzed. So this is a part of the enzyme. Enzymes are proteins. This site is composed of unique combination of amino acid residue side chains or R groups. So this particular this site, the active sites, may be composed of unique combination of amino acid and side chain or R groups. Induced fit theory, we talked about model, but let's talk about theory. Induced fit theory model proposing that binding of a substrate or some other molecule to an enzyme causing a change in the shape of the enzyme to enhance inhibit its activity. All right, so it either enhances or it inhibits, it stops the activity. Induced fit theory retains the key lock idea of a fit of the substrate at the active site, but postulates in addition that substrate must do more than simply fit into the already performed shape of an active site, must cause a change in the shape of the enzyme that results in the proper alignment of the catalytic groups on its surface. The concept is similar to the fit of a hand in a glove. The hand substrate induces a change in the shape of the glove. Although some enzymes appear to function in according to the older key lock hypothesis, most apparently function according to induced fit theory. So it's not just key lock, it's an induced fit theory, which kind of means that, like I just said, the clay and the mold, you put clay in a round mold or square mold, it takes the shape and the products are released. So that's what the induced fit theory is. It's more customized than just key and lock. It's not just one key that goes inside a lock and that's it. The key cannot change the shape. Induced fit is more customizable. Induced fit theory model proposing that the binding of a substrate or some other molecule to an enzyme causes a change in the shape of the enzyme to enhance or inhibit its activity. Induced fit theory retains the key lock idea of a fit of the substrate active enzyme, but postulates in addition that substrate must do more than simply fit into the already performed shape of an active enzyme. It must cause a change in the shape of the enzyme that results in the proper alignment of catalytic groups on its surface. So although some enzymes appear to function according to the older key, most apparently function according to induced fit theory. All right. The, the induced fit theory explains several anomalous properties of enzymes. An example is non-competitive inhibition, in which a compound inhibits the reaction of an enzyme but does not prevent the binding of the substrate. In this case, the inhibitor compound attracts the binding group so that the catalytic group is too far away from the substrate to react. A site at which the inhibitor binds to the enzyme is not active site and it's called an allosteric site. So it's not just a particular site, it can be allosteric, it can be wherever. If it sticks, then it fits. That's what allosteric means. The inhibitor changes the shape of the active site to prevent catalysis without preventing binding of the substrate, right? So inhibitor changes the shape of the active site. So active site usually, like we saw with the ES complex, it's just at one location, whereas here it's allosteric. Any part of the enzyme can be an active site. All you have to do is just tick, and that's your allosteric site. That's what it's talking about. An inhibitor can also distort the active site by affecting the central binding group. As a result, the enzyme can no longer attract the substrate. A so-called activator molecule affects the active site so that a non-substrate molecule is properly aligned and hence can react with the enzyme. Such activators can affect both binding and catalytic groups at the active site. Enzyme flexibility is extremely important because it provides a mechanism for regulating enzyme activity. The orientation at the active site can be disrupted by the binding of an inhibitor at a site other than the active site. Moreover, the enzyme can be activated by molecules that induce a proper alignment of the active site for a substrate that alone cannot induce this alignment, right? So, enzyme can be activated by molecules that induce a proper alignment of the active site for a substrate, right? That alone cannot induce this alignment, okay? Enzyme flexibility is extremely important because it provides mechanism for regulating the enzymatic activity. So how flexible enzyme is also very important. Activators can affect both binding and catalytic groups at active site, right? So that is also very important. So a so-called activator molecule. So even enzyme needs an activator molecule. Enzyme at times cannot function on their own. So they're talking about that the same molecule can also induce a proper alignment of the active site of a substrate. So there is an extra molecule that an enzyme takes help of. So that's the induced fit model. As you can see, substrate, enzyme, active site, substrate enters the active site, substrate binds, right? So substrate is binding, enzyme changes the shape slightly. As you can see, there is a significant change in the shape, right? This is perfectly customizable. So ES complex forms, right? So substrate converted to the products. Now these are two products. The products are released. Mechanism of catalysis. A mechanism of catalysis is a way in which the chemical reaction is assisted in moving forward. So how does a chemical reaction moves forward? What's the mechanism of it? Mechanism of enzyme catalysis vary, but they're all similar in that they increase the reaction rate by reducing the activations. That is the mechanism. The process of catalysis occurs in 4 steps, right? The reactor molecules get attached or absorbed on catalyst surface by chemical bonding, right? So reactant molecules are getting attached or absorbed. Absorbed reactants get activated, so there's an absorption which causes activation. Then the absorbed reactants get converted into products on the surface of the catalyst. So now they're converted into products. After the formation of product, the product molecules leave the surface of the catalyst. They say goodbye, right? So catalyst works best at an optimum pH ranges between 5 to 7 pH values. The activity of enzyme usually increases in presence of coenzyme or an activator such as Na plus or CO2. The rate of the reaction increases due to presence of weak bond which exists between the enzyme and a metal ion. Cofactor. So what is a cofactor? This is also very high yield for MCAT. A cofactor is a non-protein chemical compound or metallic ion that is required for an enzyme's role as a catalyst. A catalyst is a substance that increases the rate of chemical reaction, right? So catalyst is nothing but a substance, right? It's a substance that increases the rate of CR, chemical reaction. Cofactors can be considered as helper molecule, right? We just talked about activator molecule. Those are cofactors, all right, that assist in biochemical transformations. The rate at which this happens characterized in an area of study called enzyme kinetics, okay, which is also very important for MCAT. Kinetics is very important. Cofactors typically differ from ligands in that they often derive their function by remaining bound. Cofactors can be classified into 2 types: inorganic ions and complex organic molecules called coenzymes, which is also a very high yield topic for MCAT. What are coenzymes? Coenzymes are mostly derived from vitamins and other organic essential nutrients in small amounts. Coenzymes are further divided into 2 types: prosthetic group, which consists of a coenzyme that is tightly or even covalently and permanently bound to a protein. co-substrates and are transiently bound to the protein. Co-substrates may be released from a protein at some point and then rebind later. Both prosthetic groups and co-substrates have the same function, which is to facilitate the reaction of enzymes and proteins. All right, so they are coenzymes. Prosthetic groups and co-substrates have the same function, right? So there are two types of coenzymes, prosthetic groups and co-substrates, which typically have the same function. They work as facilitators, the reaction of enzyme and proteins. And an inactive enzyme without cofactor is called apoenzyme, while the complete enzyme with the cofactor is called as holoenzyme. So one is apo and one is holo. Apo is without cofactor and holo is complete with enzyme with cofactor. So if you have a holo, it means that it's complete. If you have an apo, it means it's incomplete or inactive. What are water-soluble vitamins? Vitamin C, vitamin B, vitamin B2, vitamin B3, vitamin B5, B6, pyridoxine, vitamin B7, biotin, and B9 folate, and cobalamin, and that's vitamin B12. And at times they do ask about vitamins in questions, and it's important that you should know which is water soluble so you can, respond to answers correctly. Effects of local condition enzyme activity. Local conditions such as change in temp and pH affect the activity of enzyme. We just read that. All right. Local conditions can affect enzyme's active site and therefore the rate at which a chemical reaction can proceed. We read that as well, right? Increasing environmental temp generally increases the reaction rates because the molecules are moving more quickly and are more likely to come into contact with each other. But we also read that if it increases a little bit too much than optimal range, then what happens that temperature increase or decrease outside the optimal can affect the chemical bonds within the enzyme and change its shape, right? So it can denature the enzyme. If the enzyme changes shape, the active site may no longer bind to the appropriate substrate and the rate of reaction will decrease. Dramatic changes to the temp and pH will eventually cause enzymes to denature, and we read that. Okay, so what is Michaelis-Menten kinetics, right? So in biochemistry, kinetics, named after Leonor Michaelis and Maud Menten, is the simplest case of enzyme kinetics, very important, applied to enzyme catalyzed reaction of one substrate and one product. It takes the form of differential equation describing the reaction rate, V, rate of formation of the product, P with the concentration rho, 2A, the concentration of the substrate A, using the symbols recommended by IUBMB. All right, so it's a formula given by Michaelis-Menten, right? So D rho by dt equals to VA Km plus A. All right, so this is a differential equation which is often written as Vmax, represents the limiting rate approached by the system at a saturating substrate concentration for a given enzyme concentration. Michaelis constant Km is defined as a concentrated substrate at which the reaction rate is half of V, so it's half. Biochemical reactions involving a single substrate are often assumed to follow Michaelis-Menten kinetics without regard to the model's underlying assumptions. Only a small proportion of enzyme catalyzed reactions have just one substrate, but the equation still applies if only one substrate concentration is varied. So reaction rate V, right, so the half, V is equal to VA Km plus A. Substrate concentration A, 0.5 V, the reaction rate V, and this is the full, so this is the half, 0.5, and this is Km, so it's right here. All right. The curve of Michaelis-Menten equation label in accordance with IUBMB recommendation. Okay. All right, so this is like a half. Okay. Lineweaver-Burk plot, very important. Lineweaver-Burk plot. Trust me, you will have at least 5 to 10 of them in biochemistry, at least on MCAT. So LBP, or double reciprocal plot, is a graphical representation used to linearize the hyperbolic relationship of enzyme kinetics by plotting the reciprocal of reaction velocity against reciprocal substrate concentration. This transformation provides a method for determining kinetic parameters like Vmax and Km through the intercepts on the axis By transforming the data, Lineweaver-Burk plot allows for easier visual interpretation of enzyme kinetics, facilitating the identification of different types of enzyme inhibition. So by transforming the data, right? So by analyzing the data, the LBP allows for easier visualization and interpretation of enzyme kinetics. So it's all about the chemical reaction, how fast, how slow, what is the reaction rate, what's the constant, where is the P, where is it differentiated, what is the threshold, where is the half Vmax that we just spoke about. So that's what it helps, okay? Which helps us identify different types of enzyme inhibition, right? So it's either inhibition or is it either activation. Competitive inhibitors, very important, cause changes in the slope of the plot without altering the y-intercept, whereas non-competitive inhibitors affect the y-intercept while maintaining the same slope. So x-intercept -1 by km, y-intercept 1 by Vmax, slope is km by Vmax. This makes the LBP a valuable tool for dissecting the mode of inhibition an enzyme might undergo, right? So this is 1 by V, this is -1 by km, this is 1 by Vmax, this is km, Vmax, right? So it kind of helps us understand, if it's competitive or non-competitive inhibitors, right? What is cooperativity? Enzymes are receptors that have multiple binding sites where the affinity of the binding sites for a ligand is apparently increased. Positive cooperativity or decreased negative cooperativity upon the binding of ligand to a binding site. For example, when an enzyme atom binds to one of the hemoglobin's four binding sites, the affinity to oxygen of the three remaining available binding site increases. That is, oxygen is more likely to bind to a hemoglobin bound to one oxygen than to an unbound hemoglobin. This is referred to as cooperative binding, right? So enzymes are receptors that have multiple binding sites where the affinity of the binding site for a ligand is apparently increased. Positive cooperativity or decreased negative cooperativity upon the binding of a ligand to a binding site, all right? So, for example, when an oxygen atom binds to one of the hemoglobin 4 binding sites, the affinity to oxygen of the three remaining available binding sites increases. Oxygen is more likely to bind to a hemoglobin bound to one oxygen than to an unbound hemoglobin. This is referred to as cooperativity. So, how cooperative the pre-existing molecule is, which will define, the affinity where, which molecule would you want to be cooperative with, somewhere that already has few molecules or somewhere else, right? So that's cooperativity. When a substrate binds to one enzyme subunit, the rest of the subunits are stimulated and become active. Ligands can either have positive cooperativity or negative cooperativity or non-cooperative at all. They don't cooperate at all. Positive cooperativity refers to a phenomenon where the binding of a ligand to one side on a protein enhancing the binding affinity of additional ligands to other sites on the same protein. This behavior is often observed in multi-subunit proteins or enzymes where the interaction between subunits leads to increased functional efficiency, right? So usually positive cooperativity also helps increase the functional efficiency. It plays a crucial role in regulating biological processes, allowing for a more responsible and adaptable system in protein-protein interaction complexes. So it plays a crucial role in regulating biological processes, allowing for a more responsible and adaptable system, right? So what they're actually doing is they're helping other, be better positively cooperative and also helping increase functional efficiency, right? So it's making the system more adaptable for protein to protein interactions and complexes. So it's pretty much of a helper. They cooperate with each other, right? Negative cooperativity means the opposite will be true. As ligands bind to the protein affinity of ligand will decrease. It becomes less likely for the ligand to bind to the protein, right? So this is positive cooperativity. Positive cooperativity. This is sigmoid, right? So that's positive. This is negative cooperative, and in the middle somewhere it's no cooperativity, right? So if you see the curve which is completely sigmoid, that's positive. Somewhere in the middle is negative, right? And this is no cooperative. There is no cooperative at all. There's nothing going on. Feedback regulation. Feedback regulation of an enzyme occurs when a product of the reaction binds to an allosteric site, right? We spoke about allosteric site, right? Wherever, the enzyme attaches, that's an allosteric site, basically, on the enzyme, and it affects its catalytic activity. Through feedback and inhibition, the cell responds to the amount of reaction product to regulate its further production, right? So feedback and inhibition is very important because cell responds to the amount of reaction the product to regulate its further production. The higher the concentration of final product, the more likely the product will bind to allosteric site of the enzyme, shutting down the pathway. This effect can be positive or negative inhibitory, right? So it either shuts down, right? And it can be a positive shutdown or a negative shutdown. Negative feedback results in inhibition of an enzyme in a biochemical pathway, reducing the activity of earlier enzymes, stopping the pathway. Negative means it's completely stopping, it reduces, and eventually just fading away. Positive results in activation of more enzyme, thus increasing its production of the product, right? Enzyme inhibition. An enzyme inhibitor is a molecule that binds to an enzyme that blocks its activity. Inhibition is honestly a very high yield topic on MCAT. Enzymes are proteins that speed up chemical reaction necessary for life, in which substrate molecules are converted into products. An enzyme facilitates a specific chemical reaction by binding the substrate to its active site, a specialized area on the enzyme that accelerates the most difficult step of the reaction. An enzyme inhibitor stops this process either by binding to the enzyme's active site, thus preventing the substrate itself from binding, or by binding to another site on the enzyme, such that the enzyme's catalysis of the reaction is blocked, right? So enzyme inhibitor inhibits the process either by binding, thus preventing substrate itself from binding, or by binding to another site on the enzyme such that the enzyme catalysis of the reaction is blocked. It doesn't matter what happens in the end, it stops it, right? So it is either at the active site or at another site, but it eventually stops the reaction. Reversible inhibitors, right? What is a reversible inhibitor? A reversible inhibitor attaches to enzymes with non-covalent interactions such as hydrogen bonds, hydrophobic interactions, and ionic bonds. Multiple weak bonds between the inhibitor and the enzyme active site combine to produce strong and specific binding. Reversible inhibitors attach to enzymes with non-covalent interactions such as hydrogen bonds, hydrophobic interactions, and ionic bonds. Multiple weak bonds between inhibitor and enzyme active site combine to produce strong and specific binding, right? So reversible inhibitors attach to enzymes with non-covalent interactions such as hydrogen bonds, hydrophobic interaction, and ionic bonds. Competitive inhibition. In competitive inhibition, the substrate inhibitor cannot bind to the enzyme at the same time. This usually results from the inhibitor having an affinity for the active site of an enzyme, where the substrate also binds, and the substrate and inhibitor compete for access to the enzyme's active site. This type of inhibition can be overcome by sufficiently high concentration of substrate that Vmax remains constant by outcompeting the inhibitor, right? So it outcompetes the inhibitor because the Vmax remains constant. However, the apparent Km will increase as it takes a higher concentration of substrate to reach the Km point, or half the Vx, right? So Km will increase as it takes a higher concentration of the substrate to reach the Km point, or half the Vmax. Uncompetitive inhibition. In uncompetitive inhibition, the inhibitor binds only to the enzyme substrate complex, so it's just ES complex. This type of inhibition causes Vmax to decrease, maximum velocity decrease because of removing activated complex, and Km to decrease due to better binding efficiency because of Le Chatelier's principle, right? If the equilibrium of a system is disturbed by a charge and one or more determining factors as temp, pressure, or concentration, the system tends to adjust itself to a new equilibrium by counteracting as far as possible the effect of the change, right? So, and the effective elimination of ES complex, thus decreasing the KM, which indicates a higher binding affinity, right? So so a system has a way of coming to an equilibrium point. It is either by taking something off or giving it in, and here it's taking off the ES complex, and decreasing the KM, which is actually increasing the binding affinity. Non-competitive inhibition. Non-competitive inhibition is a type of enzyme inhibition where the inhibitor reduces the activity of the enzyme and binds equally well to the enzyme, whether it has already bound to the substrate. This is unlike competitive inhibition, where binding affinity for the substrate in the enzyme is decreased in the presence of an inhibitor. Mixed inhibition. What is mixed inhibition? Mixed inhibition is a type of enzyme inhibition in which the inhibitor may bind to the enzyme whether or not the enzyme has already bound to the substrate but has a greater affinity for one state to another. It is called mixed because it can be seen as a conceptual mixture of competitive inhibition, in which the inhibitor can only bind to the enzyme if the substrate has not already been bound, and uncompetitive inhibition because in which the inhibitor can only bind the enzyme if the substrate has already bound. All right, so mixed inhibition is a mix of competitive and uncompetitive. A regulatory enzyme is an enzyme in a biochemical pathway which, through its response to the presence of certain other biomolecules, regulates the pathway activity. This is usually done for pathways whose products may be needed in different amounts at different times, such as hormone production. Regulatory enzymes exist at high concentration, low Vmax, so their activity can be increased or decreased with changes in substrate concentrations. Enzymes which catalyze chemical reactions again and again are called regulatory enzymes, right? So enzymes which catalyze chemical reactions again and again are called regulatory enzymes. Regulatory enzymes exist at high concentration, low Vmax, so their activity can be increased or decreased with change in substrate concentration. So regulatory enzyme, basically they have a function of regulating the pathway activity. What are allosteric enzymes? These type of enzymes present two binding sites, substrate of the enzyme and the effectors. Effectors are small molecules which modulate the enzyme activity. They function through reversible, non-covalent binding of regulatory metabolite in the allosteric site, which is not the active site. Anywhere else but not the active site is allosteric site. When bound, these metabolites do not participate in catalysis directly, but they are still essential. They lead to conformational changes in concrete part of the enzyme. These changes affect the overall conformation of the active site, causing modifications on the activity of the reaction. What are covalently modified enzymes? Covalently modified enzymes are modifications made using covalent bonds. Enzyme structures are modified to either activate or inactivate the enzyme molecule. Um, so it's the reason why they're modified is to activate or inactivate the enzyme molecules. The functional groups on amino acid form covalent bonds to molecules that are often catalyzed by other enzymes. Covalent modification are enzyme catalyzed alterations are of synthesized proteins and include the addition or removal of chemical groups. Modifications can target a single type of amino acid or multiple amino acid, which will change the chemical properties of the site. What are zymogens? A very important high yield topic. Zymogen is an inactive precursor of an enzyme. Zymogen requires a biological change such as hydrolysis reaction, right? We just spoke about different types of enzymes. Revealing the active site of changing the configuration to reveal the active site for it become an active enzyme. The biochemical chain usually occurs in Golgi bodies, where a specific part of precursor enzyme is cleaved to activate it. The inactivating piece, which is cleaved off, can be a peptide unit or can be independently folding domains comprising more than 100 residues. Although they limit the enzyme's ability, These N-terminal extensions of the enzyme or a pro-segment often aid in stabilization and folding of the enzyme they inhibit. Enzymes can exist in inactive forms and later be converted to active forms is by activating only when a cofactor called coenzyme is bound. In this system, the inactive form, the apoenzyme, becomes the active form, holoenzyme, when the coenzyme binds, right? So zymogen itself is a completely different topic. Enzymes can exist in inactive forms and later be converted to active forms, right? So zymogen is an inactive precursor of an enzyme and it can be activated after it is inactivated, so it can go back and forth, right? All right. So, whoops, what just happened? Hold on just a second.